So we have seen now how star works. We have seen how HiSet2 performs the mapping. So what about the mappers that map to the transcriptome, like Callisto and Salmon? How do they work? Well, what they do is instead of chopping the reads into seeds, like star does, Callisto and Salmon will cut the transcripts into pieces of 31 base pairs. And these pieces of sequence are then converted into graphical representations because it's much easier to align to such a graph than to align to a piece of text. This goes much faster. Let's take a look in Galaxy. So when you type here in the tools section Callisto, as you can see here, you're going to select a transcriptome instead of a genome because Callisto is a mapper that maps to the transcriptome. And again, you see that many transcriptomes are built in here. You have to tell him whether you have single-end or paired-end data. You have to define the input files. So what are your input files? Well, the clean, fast Q files, just like always. And then you have to tell him what the distribution of the fragment lengths in your library is. He is very sensitive to this parameter. It has to be exact or the mapping will not be really good. So for paired-end data, this is not an issue because he can calculate these parameters himself based on the reads. But for single-end data, you have to tell him these parameters. So the mean and the standard deviation of the fragment lengths in your library. So when it's your own data, there's not an issue. You will probably know the distribution of the fragment lengths in the library. But when it's public data, you have no idea. So unless it was published, you have totally no idea what the distribution of the fragment lengths was. So unless you know these parameters very well, Callisto will not work well on single-end reads. This parameter, the one that you see here, is linked to the counting that Callisto performs. So for RNA-seq data, you are going to do counting after the mapping. For each gene or transcript, you want to count how many reads were mapped to it, because these counts represent your expression levels, and that's ultimately what you want to analyze. So Callisto and Salmon, they both do mapping and counting, and they are mainly counters. So by default, they will produce files that contain counts, and they will not produce a BAM file. So when they count, they can take into account differences in GC content between different transcripts. So transcripts that are very rich in GC will have fewer reads because GC-rich regions are very hard to sequence. So Callisto can take that into account, and Salmon also. So that's what this parameter is about. Do you want to perform a correction based on GC content? And what is also an important parameter here is this one, the strandedness. So you have to tell him whether or not you used a stranded library. So what is a stranded library? It was explained in the e-learning system. So it means that for each read, you know whether it comes from the plus or from the minus strand. And then you have to define the output. So again, by default, he will only produce count files. If you want to see a BAM file, you have to set this to yes. It is helpful to have a BAM file if you want to take a look at the quality later on using Qualimap, or if you want to visualize the mapping results in IGV, you need BAM files. So Salmon is very, very, very similar to Callisto. It uses the same approach. It has mostly similar parameters as Callisto. It has a few specific ones. So it has a parameter which is called recover orphans. And it's a good idea to set this one to yes if you have paired end data, because it's established that it will give better, more accurate results. You need to know that the version of Salmon in our Galaxy server is quite old. And this is why the version that is now built in Galaxy does not perform soft clipping. But in the newer versions of Salmon, there is an extra parameter for that, where you can specify whether you want to do this, yes or no.